Chemical biologists like Chandra Tucker have found novel ways to analyze biological mechanisms. What we'll talk about today is how we can use light to activate proteins. In one of her papers, Rapid Blue Light Induction of Protein Interactions in Living Cells, Tucker wanted to see if light could induce movement of proteins from the middle of the cell to the cell membrane, which is on the edge of the cell. Basically, this would be like us wanting to see if we held a treat in our hands across the yard from our dog, would it know to come to us? It's a bit more complex and robust than that, though. First, let's understand the proteins involved. The first is Cry2. This is cryptochrome 2, a light-responsive photoreceptor. That means that instead of a usual ligand binding to a receptor to cause change in the protein, it's a photon of light that induces action in this protein. In the case of Cry2, it's blue light. Many cryptochromes are found in plants, which makes sense because many of a plant's functions need to be modulated by light, but Cry2 is found in humans, so it's a very interesting protein to learn more about. The other protein that this experiment investigated is called CHEM1810, and this is a protein that Cry2 binds to when it's activated by light. So this means that blue light means movement, right? But how could we be sure that this movement occurs? And how could we visualize this movement of Cry2 to CHEM1810? Thanks to fluorescent labels, we can. In this experiment, a red molecule, aptly named M-cherry, was bound to Cry2, and a green molecule, aptly named green fluorescent protein, or GFP, was bound to CHEM1810. CHEM1810 was localized at the cell membrane, and Cry2 was localized in the nucleus. So when light shines, what would we expect to see? Right, we'd expect to see the red move toward the green, and we would see it spread and move outward toward the cell membrane. And that's exactly what happened. You may be thinking, so what? That's great that proteins move and all, but why is that important? Sure, my dog comes to get a treat, but that's not spectacular. But let's think. If a fluorescent molecule could be linked to Cry2 and CHEM1810, why not other proteins? Tucker's lab thought this as well, and in lots of other experiments proved that linking two halves of different proteins to each Cry2 and CHEM1810 could induce a variety of protein activity. In addition to seeing how light can induce interaction of Cry2 with outside proteins, Tucker investigated how light induced interaction of Cry2 with itself, an interaction known as clustering. The light-dependent clustering of Cry2 has been useful to stimulate protein function, but Cry2 clusters poorly on its own, requiring outside factors. In her paper, an optimized optogenetic clustering tool for probing protein interaction and function, Chandra Tucker describes a new Cry2-based optogenetic molecule called Cry2-olig, which is Cry2 containing an E490G mutation. This mutation makes Cry2-olig better than Cry2 because it can be used on its own to probe and detect protein interactions in cells when exposed to light. Cry2-olig is used in a new optical approach designed by Tucker and her team called light-induced co-clustering, or LINC for short. LINC allows the visualization of protein interactions. In this paper, Tucker saw that by using Cry2-olig and a small molecule, calcium, blue light could induce clustering of two proteins, CAMK2 and CAM, to Cry2-olig. Tucker then used LINC with FRAP, fluorescence recovery after photobleaching, which is a method used to monitor live interactions in cells. FRAP does this by connecting the amount of fluorescence to the movement or exchange rate of the protein of interest. In Tucker's experiment, she used Cry2-olig tagged bait and fluorescently tagged prey to monitor the exchange rate of the fluorescently tagged protein. The idea of this experiment was that blue light would trap the Cry2-olig and would in turn restrict exchange of any other interacting protein. To test this, the team studied a protein we will call H in the postsynaptic density of neuronal synapses, or the space between neurons. Keep in mind that normally H would be exchanged from the presynaptic to postsynaptic density. Tucker observed a few things. First, that if Cry2-olig was linked to H and blue light was not shown, then exchange occurred normally. Then, she saw that if both Cry2-olig and another protein, DS-red, were linked to H, normal exchange occurred when blue light was not shown. Then, when blue light was shown, the exchange of DS-red H was affected, indicating that Cry2-olig clustered with itself in response to light and blocked the movement of DS-red. However, this is not always the case because this blockage did not occur in experiments with other proteins. With LINC and the LINC-FRAP experiments, Tucker and her team were able to show that by modifying Cry2, she could affect important protein interactions. She proceeded to study other specific protein interactions that are stimulated by clustering. So in conclusion, Through the careful examination and manipulation of proteins, chemical biologists like Chandra Tucker really shed some light on the world of biology as we know it.